Hello, everyone. Thanks for the organizer to give me this opportunity to present some of our recent research. So the talk is about molecular diagnosis enabled by dynamic DNA assembly and the CRISPR-Cas toolbox. I'm Feng Li from Sichuan University. Initially, our lab was actually started at Brock University, Canada. I set up the lab in 2014, and until 2019, we moved to Sichuan University in China, where we started a lab. We named it Analytical DNA Nanotechnology Laboratory. So one of the major research goals in our lab is to do research at the interface of analytical chemistry and laboratory medicine, with the goal to develop better analytical tools for in vitro disease diagnosis. So diagnosis is the first step to prevent or treat diseases. So normally, most of the diagnoses, they take places in centralized facilities, such as the labs in hospital and the diagnostic laboratories. But in many cases, those diagnoses could also take place in community or even rural areas with the analytical tools known as point-of-care testing, POCT for short. So in such cases, there are actually different levels of the diagnostic conditions. So the World Health Organization actually classified the diagnosis conditions into 4 different levels, from 0 to 4, based on the available resources, such as lab facilities and the trained personnel. So there are always test trade-offs at the different levels of healthcare systems. Essentially, there is a balance among the assay accuracy, the accessibility, as well as the affordability. So basically, to develop a diagnostic test, so here I show a typical workflow for it. So basically, we have to consider 4 essential components. It involves the collection of clinical samples from patients, the recognition and the labeling of biomarkers in the clinical samples using the diagnostic probes. And finally, with the detection and or quantification using an instrument. So one of the question is how we can develop in vitro diagnostic tests with high accuracy and high accessibility for point-of-care settings. So one of the possible solutions to develop those point-of-care tests with both high accuracy and high accessibility is through the rational design of diagnostic probes with high accuracy and low instrumental needs. So basically, our lab focus primarily on the development of diagnostic probes, which are used to recognize and label biomarkers in clinical samples. So we treat such biorecognition and the labeling processes as chemical reactions. So since they are chemical reactions, they must follow the rules of fundamental chemical kinetics and thermodynamics. So this serves the basis of the major approaches in our labs, which is the thermodynamics guide design of diagnostic probes. The goal is to achieve both high accuracy and low instrumental needs. So basically, we focus on the study of thermodynamic basis of diagnostic probes, particularly for their analytical performance. For example, the sensitivity, specificity, what are the bases for those. And then with those insights of the diagnostic probes and their analytical performance, we use those to guide the rational design of new generation of diagnostic probes. And then we make use of those diagnostic probes in clinical diagnosis. So here is an example of a very recent technology developed in my lab, which we called it a PANDA technology. PANDA is short for post-amplification SNV-specific DNA assembly. So in this particular technology, what we are developing is a new type of diagnostic probe for single nucleotide variants. The single nucleotide variants are very important biomarkers for cancer diagnosis and prognosis. So here in the bloodstream for cancer patient, there could be many of those tumor-derived single nucleotide variants. Those could serve as marker and signature for the cancer patient. However, in actual clinical samples, there are much more wild-type sequences than the tumor-derived SNV. And these DNA sequences, they're basically the same as SNV, except bearing one single nucleotide difference. So the issue is how we can develop trace amount of SNV from high background of wild-type sequences. So basically, we developed this DNA assembly technology. So the assembly process is highly sensitive to the single nucleotide mutation. So we can actually convert the traditional monotonic signal into this bimodal signature. So in that cases, we can strongly suppress the signals of wild-type sequences. And in this case, we can achieve extremely sensitive detection of SNV in clinical samples, which known as the liquid biopsy for cancer. In this case, we collect the clinical samples from 108 cancer patients, and we detect the drug-sensitive SNVs in these clinical tumor and blood samples. And we also compare our PANDA technology with the NGS deep sequencing and the digital PCR. They show highly consistency with those two state-of-the-art techniques. And the general clinical accuracy is about 95%. And we also found in some cases, for example, in patient number 81 and the number 103, in our PANDA technology, we actually find these two patients are positive for the T790M mutation. However, the digital PCR classified them as negatives. And we also compare the results with the NGS deep sequencing and confirm that those two patients are actually positive. So basically, this means our technology could be more accurate than the digital PCR. And because this is an innovation at the level of diagnostic probes, we don't rely on any specialized equipment. So we can achieve better accessibility with much reduced instrumental need and much more affordable than the digital PCR or next generation of sequencing. So this is an example of what our research looks like. So let me switch gear a little bit to the CRISPR diagnosis. So in addition to the dynamic DNA assembly or dynamic DNA nanotechnology, we are also interested in working with other systems to create diagnostic probes to enhance both test accuracy and to reduce the instrumental needs. So one of the most exciting tools is the CRISPR-Cas machinery. With these two seminal works reported by Feng Li and Jennifer Doudna, many researchers quickly realized that CRISPR is not only a powerful tool for gene editing, but could also serve as an extremely powerful tool to create diagnostic tests. So the fundamental basis for CRISPR-based diagnosis is mainly the unique target-induced transcleavage activities of Cas12 and the Cas13 family proteins. So basically, only upon the recognition of target DNA or RNA using the cRNA-Cas complexes, the indiscriminative DNA or RNase activity can be triggered and cut single-stranded DNA or RNA reporters. So let's take the Cas12a as an example, which is the core for the detector diagnostic platform. So it holds the helicase activity, so it can directly recognize a specific double-strand DNA, such as amplicons of PCR, LAMP, or RPA, without the need to convert it to single-strand DNA. As for other hybridization probes, they have to convert a double-strand DNA into single-strand DNA, either by high temperature or by other approaches. And the transcleavage activity of the Cas12a is naturally a powerful signal amplification system. So the CRISPR-Cas12a can streamline highly specific target recognition with powerful signal amplification, and is fully compatible with isothermal amplification techniques such as LAMP or RPA. So in that case, CRISPR-Cas system is a perfect platform to enhance assay accuracy, meanwhile reduce instrumental needs. So here I'm going to use two specific examples to showcase some of our own research to make use of CRISPR-Cas systems to develop assays for nucleic acids and protein biomarkers. So in the first example, we developed plasmonic CRISPR-Cas assays for viral infections. So this project is actually initiated with a collaboration with the Cool Climate, Urology, and the Viticulture Institute at Brock University. So basically, the grape and wine industry is extremely important in the Niagara region where Brock University and the Covey located. So the grapes rank as the second largest fruit crop in Ontario, Canada, which generates an economic impact of 4.4 billion Canadian dollars annually. And the Niagara region is the largest and the most important region for grapes production in Ontario. So one of the very important roles for Covey is to provide grape wine viral test for the local vineyards. So grapes, just like human, they could be infected with many viruses. So there are known more than 70 grape wine viruses worldwide. They could cause diseases such as grape wine leaf roll disease, the grape wine fine leaf degeneration complex, and the grape wine red blotch disease. So we focused on the grape wine red blotch disease. So this disease is caused by the virus known as the grape wine red blotch virus. It is a single-stranded circular DNA virus, first identified in 2008. It costs vineyards owners as much as $65,000 per acre. And the vineyards, once it has been infected over 20%, it has to be replaced. So actually in Canada, there is a national clean plant program. The goal is to promote production and use pathogen-free healthy plant material for food crops in Canada. So the slogan is start clean and stay clean. So it's very important to have a field-based test to actually can directly detect viral infections in the fields, in the vineyards. So basically the goal of our collaboration is to develop a field-based test for grape wine viral infection. So ideally, it is a nucleic acid-based test because nucleic acid-based test is one of the most accurate and sensitive tests available. However, in most cases, nucleic acid testing has to be done in laboratories rather than in field. So the idea is to combine nucleic acid amplification with CRISPR technology, as well as a color matrix signal readout system to develop a visual color matrix grip-1 viral diagnostic test that is accurate, rapid, and field deployable. So for the color matrix test, we make use of the plasmonic nanoparticles, such as DNA functionalized gold nanoparticles. So these nanoparticles, they initially are red colored because of the absorbance of the solution is at around 520 nanometer. However, if we have additional cross-linking DNA, as denoted as here, to cross-link the gold nanoparticles so that they come close to one another, then there's going to be a plasmonic coupling, and wavelengths would shift from 520 nanometer to over 600 nanometer. So the solution of this gold nanoparticle would change from red to blue. So this strong color contrast would give us a signal for the color readout. And then how we can combine it to use it as a readout for the nucleic acid testing for the grip-1 viral infection. So basically, here is a typical workflow developed by my student Yongya and my postdoc Haiyam. So we extract nucleic acids from the leaves of the grape, and we amplify them using nucleic acid amplification techniques such as PCR or RPA. Then we design a cRNA, and once it combined with Cas12a to form the cRNA and Cas12a complex, when they recognize this viral-specific amplicon, they will trigger the trans-cleavage activity and cleave this linker into pieces. So if it is a positive sample, there will be no linker, and the solution remains to be red. If there is a negative sample, so this linker would cross-link gold nanoparticles, and the solution would be the blue. So we basically collect samples in field from the vineyards, and we collect the leaves and extract the DNA on site. And then we perform the test as I just described, and we can easily find for any type of the red-berried or white-berried grapes, we can all detect the viral infections. Those are different types of grapes. And once they be infected, they all show red colors. And we also compare it with standard PCR and gel electrophoresis, and we got a very consistent result. And more importantly, even for this field test, we can dilute the initial DNA extraction solution to as much as 10,000 times. We can still visualize the color difference. So here we use a single color. Sometimes it's a little bit for us to differentiate whether it's a yellow or purple or blue color. So in that case, we further developed a dual color plasmonic CRISPR Cas12a system. In that case, we developed two color readout. And those two color readouts, they have a different mechanism. So they are orthogonal color readouts. And then we combine them, integrate them with CRISPR technology. In this case, we developed this assay for a human viral infection, the hepatitis B viral infection. So again, the typical workflow involves the amplification. So here we use a LAMP amplification, and then we design a cRNA for the specific gene of HBV. And once it combined with CRISPR, it will cleave the substrate and then trigger the color change. In this particular case, we have two types of color systems. The first color system is the same as I just described for the grape viral infection. So the second system, the gold nanoparticles was unmodified. In that case, they are very sensitive to salt. If the salt concentration in the solution is very high, so those gold nanoparticles tend to collapsed together to show the blue color. However, if there is a single strand DNA, they can protect the surface of gold nanoparticles to prevent the aggregation even in high salt concentrations. So in that case, the color will be red. So because of two color systems, so if we have a positive HBV infection, in the first system, the C1 here, the color will be red because the linker has been cleaved. But in the second system, because the single-strand DNA has been cleaved into pieces, so they cannot protect DNA, gold nanoparticle anymore, so the color will be blue. So the red and blue indicate a positive sample, and the blue and the red indicate a negative sample. So we perform this for different concentrations of HBV plasmid, and we find that indeed we have two color readouts. One thing is they can help to confirm the detection results. And the second, we can actually use the ratio of the two channels to get a quantitative readout to detect HBV with concentrations as low as 10 attomolar. So this is the first example how we can make use of CRISPR combined with plasmonic nanoparticles to develop color matrix systems for viral infections. So the second system is to make use of CRISPR to develop ultra-sensitive platforms for protein detection. So one of the reasons why we want to make use of CRISPR to do protein detection is because many important biomarkers are proteins. However, unlike DNA, those proteins cannot be replicated and amplified. So the typical limit of detection for proteins are much worse than those for DNA. So how we can detect proteins, make use of the powerful amplification power of CRISPR-Cas systems. So basically in the literature, there are a couple systems. In one system, it's called immunosystem. In that case, the protein would be captured with a capture antibody, and we label the antibody with the double-strand DNA, just like the conventional ELISA assays. This is a little bit like the immuno-PCR system, except that the detection is achieved by using the CRISPR-Cas systems, making use of the transcleavage. Another typical type work is to make use of aptamers. So those aptamers can switch their structures and trigger subsequent amplification reactions upon the binding with proteins. One of the issues of this system is that for the immunosystem, it always has very high background, like immuno-PCR. But for the aptamer system, there are not enough aptamer available for designing such assays. So we develop a system, we call it protein-responsive DNA assembly. So the idea is to, in a homogeneous solution, we directly convert proteins into DNA so that any DNA-based detection techniques can be used for protein detection. So we developed several different strategies, different generations of techniques to achieve this goal. Indeed, we can achieve the conversion of protein to DNA in free solution. And we can make use of those techniques to do like wash-free imaging of cell surface markers. And we can even combine with highly sensitive electrochemical systems to develop rapid one-step detection of PSA for cancer screening. And of course, we can combine it with CRISPR systems. So in this particular case, we integrate a protein-responsive DNA assembly. So in this case, the two probes are linked with antibodies that can recognize a specific protein. In this case, is interleukin-6, but at different epitopes. Once they bind to the protein, they would bring those two short DNA strands, the red part here, in the solution in very close proximity, and they assembled. And we can do a primary extension and to generate double-strand DNA or single-strand DNA that can be recognized by the CRNA and the Cas12a complexes, and trigger transcleavage activities. And this can push the detection limit for proteins to femtomolar level. So in the rest of my talk, I'm going to give a very specific example of how such assays can help with the clinical diagnosis. So here we use it to develop an ultra-sensitive assay for anti-SARS-CoV-2 antibodies and to make use of this assay for vaccinated kidney transplant recipients. So why we are interested in this particular group of patients? Because for this particular group of patients, once they be injected with vaccines, like 2 doses, standard like 2 vaccine doses, so only 4% to 48% of the kidney transplanted recipients have detectable antibodies, either the IgG or IgM, which means the conversion rate is much lower than normal than the normal group of people. However, there are also evidence that there are, even for those patients, they cannot detect anti-SARS-CoV-2 antibodies in their bloodstreams. There are still cell-mediated immune responses. So in those kidney transplants. So the question is whether those patients cannot develop anti-SARS-CoV-2 antibodies at all, or they can still produce antibodies, but antibody levels is too low that the standard techniques in hospital cannot actually detect those antibodies. So this is a very important question, because if those patients can indeed produce antibodies, only the level is low, we can give them booster doses. But if they cannot produce antibodies at all, there has to be different strategies to prevent this group of patients. So we developed an ultrasensitive assay by combining the protein responsive DNA assembly, here we call it a molecular translator, to convert a SARS-CoV-2 antibody into a DNA barcode. And then we make use of CRISPR system to develop an ultrasensitive assay so that it can actually recognize the protein biomarker. So here is the design. So basically, we have 2 DNA probes. So one DNA probe is linked with the RBD of the anti-SARS-CoV-2, of the SARS-CoV-2 antigen. And the other is to recognize a human antibody. So once there is an anti-SARS-CoV-2 in the sample, so the 2 affinity ligands would bind to the same antibody and assemble the DNA. And then there's going to be a primary extension reaction to produce double-stranded DNA barcodes. And then we design cRNA and the Cas12a complexes can recognize specifically with this DNA barcode and trigger the subsequent transcleavage activities. And we could also use RPA to amplify those barcodes in isothermal conditions to further amplify the detection signal. And how sensitive could this assay be? So once combined with RPA and CRISPR-Cas cleavage, we can achieve a sensitivity that is 10,000 times more sensitive than the commercial ELISA kit for both IgG and IgM. So with this ultra-sensitive assay, we're also concerned about assay specificity. So in that case, it also gave us extremely high assay specificity. It can differentiate SARS-CoV-2 from MERS or SARS-CoV, or it could even differentiate different variants of anti-SARS-CoV-2 antibodies that are against the wild type or the Delta variant or the Omicron variant. So once we confirm this assay extremely sensitive and specific, we validated with healthy participants. So there are a total of 137 healthy participants. We found that our assay is extremely sensitive, that can differentiate the patients or the healthy participant that has been injected with 2 doses of vaccine from the unvaccinated group. And it is also highly specific, give us minimum amount of false negative or false positive result. And then we make use of to test the serum level of SARS-CoV-2 antibodies in the kidney transplanted recipients. So we collected samples from a total of 85 KTRs, and we found that indeed the overall signals for the kidney transplanted recipients, their level of antibodies are much lower than healthy groups, either for the IgG or for IgM. However, the level of this KTR antibodies in KTRs are actually significantly higher than the negative groups, which means for most of those patients, they can actually produce antibodies. And the antibody level is much lower than healthy groups. And then we tracked down another 33 patients to give them the third dose and detect antibodies. We found a significant increase of antibodies in their bloodstreams. And 2 of those patients highlighted with a star here, they can even have a detectable level of antibodies by hospital-based diagnostic techniques. So in summary, I think it is possible to design analytical tools with high accuracy, high accessibility, and high affordability through thermodynamics-based rational design of diagnostic probes. And the CRISPR-Cas system is also an ideal platform to engineer diagnostic tools with high assay accuracy and low instrumental needs. And the CRISPR assays are highly sensitive and field deployable, which will find wide applications in diagnosing both human and crop diseases. So with those, I would like to thank all my students, the postdocs, and the collaborators from various hospitals and the universities and funding agencies. And thank you for your attention.